Hello, Growth Leaders listeners. I'm thrilled to welcome our distinguished guest today, Mr. Mike Rossi, the Vice President of ConcertAI, a trailblazer in the field of oncology and data research. In his remarkable career, Mr. Rossi has not only spearheaded groundbreaking initiatives to eradicate cancer through data-driven approaches, but has also played a pivotal role in ConcertAI's ascent to industry prominence. Today, we'll unravel the mysteries behind its research journey, explore ConcertAI's game-changing innovations, and discuss the profound impact on the global landscape of cancer research. But this episode is not more than just a glimpse into the professional journey. It's a peek into the person behind the title. Join us as we delve into personal insights, discuss Mr. Rossi's vision for the future of oncology, and discover the strategies behind maintaining a healthy work-life balance. Get ready for an insightful conversation with a true growth leader, Mr. Mike Rossi, here on Growth Leaders by Short Learning Solutions. Welcome, Mr. Rossi.
Thank you very much. Happy to be here.
It's great to have you on the show. So before we start, what is important in your life, Mike?
What is important in my life? My family, my 3 children, my wife. Curiosity. I have tremendous scientific curiosity. So the world around me continues to fascinate, inspire me. So that's really why I'm a scientist. That's why I do the work that I do, is because I'm constantly fascinated by health and how health turns into disease and what drives disease.
Yeah, and that is super interesting. I mean, it's something I can mean that there's so many parameters in there and trying to understand how those interact with each other and so on. So tell us a little bit background about your journey in oncology and data research, and if there has been some pivotal moments during that journey, maybe before or after, or when you entered with the ConcertAI too.
Yeah, it's an interesting story, I think. Um, as a child, my uh, maternal grandfather, so my mother's father, he was a physician at a time when family practice, family medicine was really in. And then as I got older and I went to college, I thought I wanted to pursue medicine, but I didn't enjoy the patient interaction side of it. I really was much more interested in the science, what drove disease. And so that led me through an interesting journey where when I initially finished my undergraduate degree, I started working for Roche Biomedical Labs, which was a division of Hoffman and Roche. And at the time in the early 90s, the only genetics we really had was cytogenetics. And I was just very lucky that my career trajectory kind of followed technology. So I started when the first kind of molecular based technologies, FISH, fluorescence in situ hybridization, came about. And then I was encouraged to go and get a PhD, and I did. And I did my PhD at the University of West Virginia because we had an affiliation with Lawrence Livermore Labs. And Lawrence Livermore, it was at the time of the Human Genome Project. They were sequencing chromosome 19. And I had done so much FISH work that I thought I was going to be involved in helping to map some of the genes on chromosome 19. And I just had a wonderful PhD mentor, Don Sens at West Virginia, and he was very adamant that I not just be a molecular biologist, but that I understand cell biology as well. So I did electron
What is it?
microscopy. Yeah.
What is the chromosome 19? Is there a name for that,
Oh,
or that is just chromosome 19?
yeah. So every healthy individual has 46 chromosomes.
Uh-huh.
Men have an X and a Y, women have two X chromosomes, but then you have 22 autosomes. So you have chromosome 1 through chromosome 22, and so chromosome 1's the biggest, and chromosome 22 is the smallest. So
Uh-huh.
19 is a smaller chromosome. It's not quite as big as 1. But yeah, after I finished my PhD, I went to Roswell Park Cancer Institute. because that was the basis of the Genome Project. So they got one
Mm.
person's DNA and they chopped it up and they put them into what are called bacterial artificial chromosomes. So they took stretches of human DNA and they put them in these bacterial constructs so that they could grow up many copies of the DNA. So I was very
Okay.
lucky because I was at Roswell Park. We had the genome in a freezer and we started to develop what are essentially the first copy number arrays. So we are actually able to take those backs that were used for the Genome Project and then look for regions of gain or loss within individuals, especially in a cancer context. But it was also applied to rare disease as well. So patients with idiopathic conditions, poorly diagnosed conditions where chromosomes didn't have quite the resolution to be able to see smaller changes. We
Mm.
started with back arrays, and then all that turned into eventually sequencing, and that was the next part of my journey. Was I wound up at Yale, and I was at Yale to do a fellowship in molecular genetics and cytogenetics. So I got a laboratory director training position at Yale, and Yale was essentially where kind of the first commercial whole genome sequencing happens. One of the first projects was to sequence an individual called a hat map. We had these kind of
Uh,
human genomes that we had in store
uh,
and that we were
and
using to study individuals.
what would you use those mappings to? Those sequences?
Yeah,
You
and so
do
that
that
was the
so
whole idea.
that yeah.
So that changes, any changes in DNA, especially within genes. So there are 3 billion nucleotides. So there are 4 nucleotides, A, T, G, and C, that make up DNA. And any change, so an A to a T or an A to a G or an A to a C that happens in a gene can change biology. So it could be the difference between whether you have blue eyes or green eyes, but it could also mean the difference between having a certain type of cancer that's more aggressive or less aggressive, depending on what mutations happen and what specific genes are mutated. So changes in DNA that happen in individuals prior to birth. So when you have a change in sperm
Mm.
or egg, that leads to underlying genetic conditions, some that you see at birth, some that you see later in life. But then there are also changes that happen as cells are just dividing and as you're just living your life. And that's really where the cancer process comes from. So what's really fascinating just through the breadth of my career is understanding how it's not just the genetics, but it's how all the underlying biology, how our immune system is related to changes in those cells
Yeah.
and where those cells happen specifically in the body. Uh,
And that is, and I fully understand you need a lot of experience just to starting interpreting, and then even more experience to start to draw conclusions in there.
right.
And we gotta try to find out a little bit more here. So in the realm of oncology, so what aspects and specific area are you most passionate about today?
Yeah, so that's the interesting thing. So I could tell you in about 2008, there was a global economic downturn, led to a huge amount of investment in genomics. So there was actually some money that became available, and particularly in lung cancer. And we were starting to understand specific changes in genes. So, like the EGFR gene gets mutated in a specific type of lung cancer, non-small cell lung cancer, lung adenocarcinoma specifically. And we knew that patients who had specific mutations in the EGFR gene would be responsive for the first generation of therapies related to EGFR mutations. So that led to what was called the Lung Cancer Mutation Consortium. And we were doing just very rudimentary type testing. We would only look at, you know, uh, 20 different variants, so specific nucleotide changes in about 5 genes. And then
Mm.
that eventually evolved into us going from the earliest, it was called capillary sequencing, to next generation sequencing. And now we can actually sequence whole genomes, whole exomes, the exomes, the coding region, just the genes, the 20,000-ish genes that make up the human genome, protein coding genes. And that led to the biology of progressing the cancer. And initially, we thought every tumor would be sequenced. We would find specific mutations that made that tumor. And what we eventually learned that there were many tumor types that didn't necessarily have drivers. So there were other types of lung cancer, squamous cell cancer, not as opposed to adeno, it has mutations in P53. There's lots of genomic instability, but no real driver, no KRAS, no EGFR. So it became much harder. And those, then we started to look back at the biology and say, well, maybe we can't find a specific gene in the tumor, but maybe it's some other associated biology. And that's where the revolution with immunotherapy came in. And we really started to develop drugs that could turn the immune system back on and then use the immune system really to fight the cancer.
Yeah. No, just thinking here, does every person carry genes for having cancer, but they may be hidden? Because sometimes you could reflect on people who, I mean, maybe somebody died very close to you, and then that they go down, and then a short time after they actually get the cancer, which it seemed as if there was a connection. I don't know,
Right.
but
So it's very complicated. In the genetics world, we call it multifactorial. So your underlying susceptibility to certain types of cancer, breast cancer, colon cancer, about 10% of patients will have some mutation that makes them susceptible to that cancer. But that means 90% of patients with breast cancer, colon cancer, other types of cancer, there's no connection. Lung cancer, it's very rare that you have what we call a germline mutation, something you're born with that leads to that cancer. Almost all of the the vast majority of cancers are some type of exposure to life. So it may not just be ultraviolet light or smoking. It could be, having, free radicals. So when you eat bad food or bad quality food, you can develop a lot more changes, a lot more chemical changes, and some of that breakdown of those products that can lead to mutation. But all of that is connected because it's a system. It's not just one thing. And that's always what's difficult to explain to people, because there's always this assumption. My grandfather had prostate cancer and died of prostate cancer, but he may have gotten prostate cancer when he was 80 years old. You're going to see cancers happen more frequently in older individuals because patients they'd have a relative that was young and got the cancer and died of the cancer, and then they had other relatives, sometimes themselves, who got the same cancer. The vast majority of cancers, it's not that way. We still don't have a very, very good understanding of all the nuances of the germline. So some of this is genes control your immune system. So you have genes that make your T and B cells, subtle changes in those genes. And then we have other genes that metabolize everything. They break down everything that we consume, everything that goes into our bodies. And subtle changes of those genes can mean the difference between whether or not you metabolize a drug. So some people can get some people can get Tylenol or acetaminophen. Some people can break it down, some people can't. And that's a lot of times that's true of genetics.
So if we then move your career to entering the data analysis side, and I think you had something even before at ConcertAI, but now you're VP at ConcertAI. First of all, what do you guys do more than gather data, and how does that incorporate into your knowledge base which you
Sure.
have? Because I'm quite interested
Sure.
in that aspect.
That's I've been here since March of 2023, and ConcertAI is truly a data company. And my role as VP of Translational Science, and it's a long title, it's multimodal real world evidence. The multimodal real world evidence means that my role is how do we start to take multiple data types? So it's not just the genomics, it's the clinical information, surgeries, therapy, radiation, chemotherapy, other types of targeted therapies, as well as did the patient have adverse events related to those therapies? And now it's incorporating things like radiology and digital pathology. So we have this revolution in pathology now where we're able to, so traditionally the way a tumor is diagnosed is the specimens taken from the patient, and a pathologist looks at that tissue under a microscope. We're able to turn that microscope image into a digital image now, and now we can train algorithms. So machine learning and AI algorithms can be used to identify features, subtle features that are visible in the slide that a pathologist may not necessarily see or recognize. So pathology is based off of pattern recognition. So the
Mhm.
shape of the nuclei, the shape of the cells, the staining intensity of the cells tell a pathologist it's this specific tumor type as opposed to a different tumor type. Um, we can now use AI to train with those images to find the same thing and actually find more. So now we can actually see changes of the cell that tell us this patient has a fusion, a specific type of genetic rearrangement that may be responsive to drugs.
We're starting to
Radiology,
see we're starting to see a cause there
right?
uh, in those
And that's
patterns.
yeah, and that's the beauty of it. So then you have radiology. So any imaging that's done, MRI, PET, we can see what we call hot or cold tumors. We can actually see if a tumor is metabolizing at a high rate, and that
Mhm.
tells us that the tumor you can kill a tumor, right? And so what you
Mm.
want to see is the tumor, responding to the therapy and cells dying, and you can see that in radiology. So a lot of this now is how do we integrate all those really at one time disparate data types? So a radiologist did radiology, a pathologist did pathology, the genomics people did genomics. How do we start to integrate all those data types together with the clinical information to best understand how a patient goes through this journey? And using prostate cancer as an example, so you'll have patients that get diagnosed with prostate cancer, and then they go through these patterns of resistance to therapy. So we call it biochemical recurrence. So they initially appear to respond to therapy, but then they develop resistance, and then it moves to really aggressive stages, castrate-resistant prostate cancer. So you can remove the prostate, but the patient still continues to manifest disease. And how do we start to understand that entire journey? You can't understand that entire journey unless you have all the data together in one central source. And that is the data that we're going to build the future of AI models from. So all of the large language models, all the AI type technologies require very robust data sets. They have to have complete information. So we need to understand specific variables at specific time points.
Yeah. Is there any areas, tumors or cancers, where you see where you are approaching a different way of treatment than it has been maybe over the last 10, 20, 30 years? Any specific, you say, here's actually progress due to, it could be the pharma company, it could be, I mean, your and your team's input and so on, where you try to approach it in the new way, if I put it like that.
Yeah,
Uh,
yeah,
is there any
yeah.
areas you can pinpoint where it has been a breakthrough, or where you believe there are breakthroughs as such the coming years?
Yeah. So I could tell you that in lung cancer, for instance, so my career has tracked the small molecule revolution. So the first
Yeah.
generation of tyrosine kinase inhibitors, the first EGFR inhibitors came out as I was training. And I've been able to track, the progression of the first generation, the next generation. Some of those drugs have tremendously increased lifespan of patients. So predominantly in lung adenocarcinoma, so that histology, the squamous type tumors are different. So even though it's lung cancer, there are different types of
Mm.
histologies that have different response to therapies. Immunotherapies have been tremendously wonderful new addition to the treatment regimen. That's about roughly about 10 years old. Now I actually
Yeah.
see ADCs, so antibody drug conjugates, so new ways to target um, gene expression where you're using essentially an immune style targeting methodology. So you're using an antibody to identify a cell that's expressing a specific type of gene or protein, and then using different chemotherapies and cytotoxic drugs to identify just that cell, and then inject the toxin just into that cell. So antibody drug conjugates are amazing. There are other types of drugs called BiTEs, bispecific T-cell engagers, things like multiple myeloma, other types of cancers where you now can actually use that technology to identify a specific cell type and then engage the immune system to come and kill that cell. So those
Yeah.
technologies continue to evolve. We have protein degrader technologies where you have whole new mechanisms of trying to identify. So I feel like we still have a very long way to go, but in the span of my career, I've seen breast cancer patients where we know there's subsets of breast cancer patients that we just don't treat. We don't have to treat because we know that they're going to live long, full, productive lives, and chemotherapy is actually going to provide no benefit. So the only way
Mhm.
you get there is with data. So
Yeah.
cancer is really a data problem. Disease is a data problem. And so the more we can start to put together these pictures to understand as new therapies are coming
Mm.
on, how are patients responding to those therapies, identifying subcohorts. So genomics is one of the easier ways for me to
Yeah.
separate groups because we can understand a little bit more granularity of how those patients are different. And
So
yeah,
now we
I
move.
feel like, yeah.
So if we move us to 2034 now, 10 years ahead, have we, because there's long, as you say, there's the understanding, and then we have the pharma, the testing,
Mhm.
and so on. But in coming 10 years, do you see new ways to treat or delete some parts of cancer out by
Yeah,
people?
hopefully. I mean, if you look at pediatric cancers, in the 1950s and 60s, a pediatric leukemia diagnosis was lethal. Most children didn't survive.
Yeah.
Now, the vast majority of those 90% of patients will go on and live long, productive lives. There's still many cancers that are really bad. Pancreatic cancer, ovarian cancer, small cell tumors, brain tumors. I did a lot of my research work in brain tumors, particularly glioblastoma, which is a very aggressive tumor type. Lots of pediatric tumor types where we just haven't made much progress at all. And some of that has to do, brain tumors are difficult because we have the blood-brain barrier. So you can make a
Mm.
drug, but it may not actually penetrate into the brain. Um, and the uptake of those cells, human beings shouldn't get brain tumors. Those cells aren't
Oh.
dividing. But it happens, and it can be very lethal. And so my hope is that in those tumor types that we haven't made a lot of progress, particularly brain, pancreatic, some esophageal tumors. There's some really highly aggressive tumor types that some of the pediatric brain tumors where we haven't done much, and some of it's because the tumors are rare. So we don't see
Mm.
them as frequently as breast cancer or prostate cancer. But my hope is, and I think the hope of every person in this field, is to try to extend life and quality of life as best we can. So I do not have any expectation that we'll cure cancer in 10 years. But I do hope that we continue to make this upward progress. And we've made, a lot of people will say, we've made no progress in oncology. We've made tremendous progress in oncology. There are
Mm.
people alive today that are on Keytruda, which is a type of immunotherapy. 1 million patients were treated last year. 2 million patients will be treated this year. There are many people who are living to see their kids graduate from college and get married and do things that 10 years ago did not happen. And my hope is that in the next 10 years, there are
Mm.
more and more of those cancer types and more of those patients that we can help extend their life and quality of life. Cure is, we've been trying to cure since the war on cancer, which was the Nixon
Yeah.
administration. So I'm sure there are many people in your audience that don't even know who Richard Nixon is. But, that was in the
Mm.
1970s. We've made
Exactly.
tremendous progress, but there's still a long way to go.
Yeah. And how do you see distribution of treatments around the world? I got a question here before. I mean, we have a lot of remote countries also where we have the same
So
diseases
it's,
and tumors and
yeah,
cancer, etc. Where are the limitations here? Is it skills transfer or where? Because
yeah,
it's an
yeah.
important question.
Yeah, it's an important question here as well. So we have just as many
Yeah.
disparities within the U.S. So um, we're a wealthy country. We have tremendous resources, but they're not distributed equally across the country. One of the things that I'm really proud of that ConcertAI does is that we have a strong initiative in disparities and health outcomes. So understanding why specific patients don't get access to testing or therapies is critically important to our mission. And the idea is that trying to understand the healthcare system within the U.S. Um, hopefully will help us then leverage what we do outside of the U.S. So we have a strong footprint in India, and a lot of our technical
Mm.
people are based out of India, ConcertAI. That's a huge, opportunity to
Mm.
identify patients in India that can be have better access to care, better access to therapies. Um, there's no reason why we can't do this globally. It's
Mm.
just a lot of this is, it's government. It's how do you actually motivate and make resources available. It's other industries as well. So the fact that we're building, billionaires are building rockets just to go into space seems silly to me when we have so much disparity in the world. A lot of that effort, could be pretty small investments, less than $1 billion investments, could put therapies or technologies in the hands of some of those communities around the world, even
Mm.
in developed countries that don't have access to care. So this is something where the hope would be that there's better distribution of resources throughout the world, and
Mm.
unfortunately, that has been a problem we've had from the beginning.
Mm.
So we'll continue to have those problems, but yeah, hopefully, if we have better knowledge. So my goal is that, especially at Concert AI, is to use this understanding of the clinical picture of a patient, this patient journey,
Mm.
to really understand populations that we're seeing in the U.S. and how that can best be extrapolated to any population.
Mm. I see this. I mean, this is a different but a true leadership role for which has so much impact to many fields and to populations. If we look now at the younger generation, and I'm certain there's a lot of people listening into this, maybe parents, etc., and they talk with their youngsters. Maybe this is a field which is super interesting in the next 20, 30, 50 years moving forward. What should they look for getting into the research side, the science side, etc.? I have
Yeah,
too little
that's a great
knowledge
question.
to frame it more than that. So,
No, that's
but
fine. That's fine. So I could tell you that artificial intelligence is going to hopefully make our jobs more efficient. So there'll be a lot more need for specific domain expertise, and then AI will support that domain expertise. AI is going to it's going to continue to democratize knowledge. It's going to make knowledge much more readily available. So the advice I always give is develop domain expertise in something that you know and you're good at. So a lot of times people will say, do what you love. I'm always going to recommend do what you're good at. So what your strengths are. So identifying those strengths is going to be critically important because a lot of the manual tasks underlying, so a lot of administrative assistants, every executive had an administrative assistant, but a lot of those functions are now covered by technology. A lot of repetitive work, a lot of things that required an individual to move something from one place to another, that's being eliminated. I think that AI and the technologies that we're developing are going to continue to drive that to make it more efficient. So it's really identifying something that you're good at. And I always feel like in my role, it's working to people's strengths. So trying to identify what people are good at and help them continue to steer their career in that area of their domain expertise and their personality. So some of it's not just what you know, but what you're good at, and identifying those qualities are critically important for success. I think it's very hard to be successful if you don't know what you're good at, and
yeah,
hopefully what you're good at you like. I love what
yeah,
I do. I really love what
yeah,
I do, and I love what I do because I'm constantly fascinated. I'm constantly learning. There's so much more that I don't know than I know, and that's what drives my my daily
mm.
interaction, and that's the advice I
So
give to my kids.
yeah,
So
so that is really coming back to starting to understand yourself, see where am I good at, where
right,
is my curiosity, which drives the motivation for you,
right,
and there you got the formula to really excel in really any
exactly,
area, if it's building
exactly,
cars or electric cars or
exactly,
in your field. I must
right.
say, super interesting, Mike, talking to you.
Yep.
And I would like to thank you so much for being part of the Growth Leaders podcast and
Sure.
you and your team. We will be happy to follow you the years to come. Thank you so
Great, thank
much.
you, thank you, really appreciate it.
Thanks.
Thank you.
Thanks.